get Amy wired up here.
There you go. We get two.
For our next speaker. Just a reminder, if you haven't done so already, the food safety workshop, I informed you incorrectly earlier. I said it was going to be here, and it is going to be here. We're going to be rebroadcasting here at WSU Northwest Rec, but that workshop will be hosted in Prosser as the main site. So we'll get to be an external site for that one. And I'll be your host for that as well. So look forward to seeing you all here for it. All right, without further ado, please help me in welcoming Amy Masinga.
Okay. What else to add after the great talk from Lisa? In fact, we will try, and let's say my talk will be articulated around three main points. First, I will try to guide you through the nitrogen cycles and the impact applying nitrogen sources as fertilizer to the soil will have on soil chemical properties and how changes in these chemical properties can affect berry yield, particularly in blueberry. Second, I will going to present some results highlight from a long-term experiment conducted at our research station in Agassiz in British Columbia. And in a few words, we will have a take-home message, very few, one or two main points. And now, this is roughly the nitrogen cycle. It is complex with many arrows, many boxes, many words, many chemical formulas. But let me guide you through it. First of all, when we look at the nitrogen cycle, what is important to understand is the reservoir of nitrogen is the atmosphere. And nitrogen will get into the field system through several reactions or processes. The main process is fixation of nitrogen. This can be made through industrial mechanisms of the Haber-Bosch process. That's what and the Haber-Bosch process will lead to the production of chemical fertilizer. And the two main forms of nitrogen or ion we will be targeting will be either ammonium or nitrate. Then nitrogen from the atmosphere as a gas will be converted into ammonium and nitrate and wrapped up as a fertilizer. The other process is a biological process, which is mainly driven by leguminous crops. They have this ability to fix nitrogen from the atmosphere through a symbiotic relationship between the legume plant and some bacteria, the bradyrhizobia present in the soil. But we also have other sources of N input into the soil. These will be N stored in plant material, then the biomass, or N nitrogen stored in animal residues. These are the main sources of input into the soil. Now, once nitrogen gets into the soil, it can undergo several transformations. One transformation is the biological transformations. And the biological transformation will mainly be driven from and coming through the symbiotic nitrogen fixation. Then once that nitrogen is gets into the soil system, it will be transformed through microorganisms. Then that form will be mainly organic nitrogen, and it has to be transformed into inorganic or mineral nitrogen, ammonium nitrate, before it can be taken up by growing plants. Another process will be the chemical process and the physical process. These are two complementary transformations. We started with biological process. Once the nitrogen gets into the soil systems, then it will be transformed by microorganisms, and we will mainly end up with ammonium, and ammonium later on will be converted through nitrification into nitrate, and nitrate can be taken up by plants. As we saw from Lisa's talk, for example, raspberry will prefer nitrate, while ammonium will instead prefer, blueberry will instead prefer ammonium. Now, once ammonium and nitrate found themselves into the soil, they can be taken up by plants when they are into the soil solution. And once they reach the soil solution, they can be fixed through clay particles, or could be also transformed into ammonia and lost into the atmosphere, or nitrous oxide and lost into the atmosphere. This brings us to the losses of nitrogen from the soil. As we saw, there are three main sources of losses: ammonia through volatilization, nitrous oxide emissions, or nitrate leaching. Let us focus a little bit into the chemical transformation of one main fertilizer used in blueberry plants. This is ammonium sulfate. Ammonium sulfate, because blueberry will have a preference for ammonium against nitrate. Then once ammonium sulfate is applied to the soil, it will be transformed and solubilized into ammonium or sulfate. Let's talk about sulfate, for example. Once sulfate reaches the soil solution, it is not mainly taken up by plants, then it will be accumulated, and through a long period will lead into salinity. And salinity is a result of salt accumulation, and this can inhibit plant growth. Now, ammonium, once it is in the soil, it can be taken up by plants. Blueberry will prefer ammonium. But what's happened when ammonium accumulates? That means we apply too much of ammonium sulfate. Then the ammonium left into the soil at the end of the growing period or season will be transformed into nitrate. And during this transformation, there are two hydrogen ions or cation that will be released. And the release of hydrogen cation into the soil will change the pH, the pH of the soil, and lead into acidity. As we saw from Lisa's talk, blueberry prefer a range of soil pH between 4.5 and 5.5. What does that mean? It means if hydrogen cation accumulates in the soil, they will further produce acidity and lower the soil pH below the threshold of 4.5. And this can also inhibit plant growth and yield. These are some basic. Now, how does that apply in the real life? Here, I want to present results from a long-term blueberry experiment. And these results were submitted to the journal Sciences Horticulturae blueberry yield and soil properties response to long-term fertigation and broadcast nitrogen. The long-term experiment was established in 2008 following a factorial randomized complete block design. The two main factors were application method, broadcast, and fertigation. And the nitrogen rate varied with the age of the plant. For young plant, we had full rate of nitrogen, 0%, 50%, 100%, and 150%. 100% here means the recommended rate of nitrogen fertilizer at that particular stage. And for mature plant, we also had 0%, 100%, 150%, and 200%. Here, the combination of these treatments yielded 9 treatments. First of all, one control, that means 0 and application. Then broadcast 50, third 50, broad 100, and third 100. We also had broad 150, its correspondence, third 150, broad 200, and third 200. From that long-term experiment, we collected soil samples in 2013, 2014, and 2015 at 2 depths, 0.30 and 30 to 60 centimeters. In 2015, to check some observation we made, we also collected the layer of sawdust on the soil beneath, 0 to 30 centimeters. And berry yield were collected between 2010 and 2015. And in the lab, we were able to analyze from the soil and sawdust samples ammonium N, nitrate N, soil pH, and electrical conductivity. pH being the indicator for acidity, and electrical conductivity, the indicator for salinity. From the berry yield, we assess the N content and the nutrient use efficiency. Now, let's go through some data. What did we observe? Measuring the residual or post-harvest ammonium in the soil profile, we found that when there was a high residual ammonium N accumulating in the 0 to 30 centimeter and under the fertigation treatment, mostly when we applied N rate above the 100% or the recommended rate. Then applying ammonium sulfate through fertigation at a rate above the recommended rate, then we increase or we accumulate post-harvest ammonium into the soil, mainly in the 0 to 30 centimeter. In contrast, when we look at the 3 rates applied by broadcast, we did not observe any effect of N rate. Then there was no accumulation of ammonium and in the 0, 30 or 30 to 60 centimeter. Now, let's look at what happened with nitrate. In this graph on the x-axis, we have the treatment, our 9 treatment, and on the y-axis, we have our nitrate concentration. And in red, we have nitrate in the 0 to 30 centimeter, while in green, we have nitrate in the 30 to 60 centimeter. What did we observe with post-harvest nitrate in the soil profile? We observe an increase in nitrate concentration with increasing N rate, particularly when nitrogen was applied through fertigation. But the extent of increase is greater in the 0-30 centimeter compared to the 30 to 60 centimeter. What does that mean? It means when we use fertigation to apply ammonium sulfate, we accumulate nitrate in the 0-30 centimeter and to some extent down to the 30 to 60 centimeter. Again, with broadcast, there is no accumulation of nitrate as we've seen with ammonium. We were then interested in understanding why there is no accumulation of either ammonium or nitrate when N fertilizer was applied through the broadcast. Then we went and sampled the sawdust layer above the soil. What did we observe? Under the broadcast application method, we see a high amount or concentration of ammonium, and this concentration increase with increasing N fertilizer. While in the third treatment, the concentration also increases, but to a much lower extent compared to broadcast. Remember, in this study, fertigation was used as drip irrigation. Then what it means? It means once we apply, we mix the fertilizer with water, and we apply the fertilizer by fertigation, it is soluble. Then it moves through the layer of sawdust. But under broadcast, we only apply the fertilizer on top of the surface of the sawdust layer. And there is irrigation, drip irrigation. But it looks like the amount of water brought by irrigation or rain is not enough to dissolve the granular ammonium sulfate and help the ammonium to move down the soil profile. Then it stays and accumulates in that layer. Now, when we were looking at the nitrogen cycle, we saw, okay, changes in ammonium nitrate into nitrate can affect the chemical properties of the soil, particularly soil pH. And we also saw accumulation of sulfate can lead into increases in salinity. Now, how do our management practices influence these chemical properties, pH and electrical conductivity? Here, I want to present the data of soil pH in 2013, particularly in the 0-30 centimeter and 30 to 60 centimeter. What we can observe is when we look at the red bars, particularly under fertigation, we see a decreasing trend. We see a decreasing trend of soil pH with increasing N rate applied by fertigation, particularly in the 0-30 centimeter. This corroborates the knowledge of the chemistry of ammonium, then the transformation of ammonium into nitrate, which liberates H+ cation, which is an indicator of the salinity, of the acidity of the soil. When we look at the broadcast application, we don't see any decreasing trend with increasing N rate, but what we see is a decrease in soil pH in the 0 a drop of soil pH in the 0 to 30 centimeter compared to the 60 to the 30 to 60 centimeter. This means by applying ammonium sulfate through fertigation, high rate could lead or will lead into decreasing soil pH or increasing soil acidity. Now, how about electrical conductivity? Once more, in the 0 to 30 centimeter, and when we focus on this area of our graph, we see an increase in electrical conductivity or an increase in salinity with increasing N rate applied through fertigation, particularly in the 0 to 30 centimeter. There is also a trend with the broadcast application, but the extent is much more lower compared to what is happening under the fertigation. Now, we focused on the chemistry of the soil, how changes or the nitrification process, which leads into the transformation of ammonium to nitrate, can affect soil properties. Now, do we have data to confirm, yes, there is a direct link or relationship between residual ammonium or nitrate into the soil with the changes in soil chemical properties, soil pH or electrical conductivity? In this graph, we present the relationship between soil pH and residual or post-harvest nitrate into the soil. On the x-axis, we have nitrate and concentration. And on the y-axis, we have the evolution of soil pH. I want to draw your attention on the blue and green mark. The blue is fertigation at 0 to 30 centimeter, and the green is fertigation 30 to 60 centimeter. What we did observe is there is a non-linear relationship between the concentration of nitrate and the evolution of soil pH under fertigation in both 0-30 centimeter and 30 to 60 centimeter. Then yes, the transformation of the nitrification process, which leads into the transformation of residual ammonium into nitrate, releases H plus cation, and this affects the pH of the soil or the acidity of the soil. Now, how about electrical conductivity? On this graph, on the x-axis, we have nitrate N concentration in the soil. And on the y-axis, we have the electrical conductivity. Once again, when we focus on the blue and the green mark, we can observe a non-linear relationship between the concentration of nitrate in the concentration of residual nitrate in the soil and the evolution of electrical conductivity in both 0-30 centimeter and 30 to 60 centimeter. Now, how all these changes will affect the berry yield, which is the parameter of interest. On these two graphs, I want to present the evolution of the berry yield between 2010 and 2016. On the left side of the graph, we have the data from the fertigation. And on the right side of the graph, we have the data from the broadcast. Now, let us focus first on the yield of the young plant. That means the yield collected between 2010 and 2012. Between 2010 and 2012, we actually see an increase, higher yield, high berry yield from the fertigation compared to the broadcast. And the difference is between 15 and 19%. That means at young age, when we apply ammonium or N fertilizer through fertigation, we have higher yield compared to broadcast. Now in 2011 and 2012, here we start to see a differentiation in terms of berry yield between in dark color the control and the green, the red, and the blue, respectively 100%, 150%, and 50%, 100%, and 150%. We have the same trend here on the broadcast in 2011. And in 2012, in both application method, we have the same trend, lower yield under control compared to the other N rate. But the striking part or the more interesting part of this result is during the period between 2013 and 2016, when the plants are somehow mature. What do we observe? Let us first, okay, the blue color, as we said now, is the 100% N rate. The red color corresponds to 150%, and the green color corresponds to 200% of the N rate applied. We see high yield under these N applications compared to the control. But when we move in 2015 and 2016, we see a drop in berry yield with increasing N rate, particularly under fertigation. And we estimated that between the recommended rate and the 150% N application or N rate, we have a drop of 19% of berry yield. And between the recommended rate and the 200%, we have a drop of 30% of berry yield under fertigation. That means by applying high amount of ammonium sulfate through fertigation compared to the recommended rate, in the long term, we run the risk to see our yield decreasing. And we explain that this is mainly due to the changes that occur, to the chemical changes that occur in the soil. In contrast, when we look at the broadcast application method, there is no changes in terms of differences between the N applications rate. Now, yes, we know there are changes, there are chemical changes in the soil. We are also seeing decreasing yield with increasing N rate when we apply by the nitrogen fertilizer through fertigation. But is there any direct link between these two changes? Okay, before going that, we're also interested in the fruit quality attribute. As Lisa mentioned, we have we also observe decreasing in fruit size under fertigation and decreasing in fruit size with increasing N rate. Now, let's see how changes in soil properties will affect the berry yield is there any direct link between the two? On this slide, we have on the x-axis electrical conductivity, and on the y-axis, the evolution of berry yield with time. We only focus on the 0 to 30 centimeter, and where we measured electrical conductivity or salinity. And once again, I want to draw your attention on the blue and the green mark. What do we see? These correspond to, the blue mark corresponds to the 0 to fertigation in 2013, while the green mark corresponds to fertigation in 2014. Then we pull the data, the yield data from 2013 and 2014 on the data of salinity or electrical conductivity from 2013 and 2014. What do we observe? Under fertigation, we have a linear relationship between electrical conductivity or salinity and berry yield, but that linear relationship has a negative slope. What does that mean? It means as electrical conductivity increases in the soil, then there is a direct effect on berry yield, particularly under fertigation. Under broadcast application, this is not being observed. And using this linear relationship, we were also able to set a critical electrical conductivity level, which is 760 microsiemens per centimeter in the 0 to 30 centimeter. That means above this critical EC level, we know there will be changes in berry yield. Now, what is the take-home message from these results? What we can say is long-term fertigation of ammonium sulfate at rates greater than the recommendation can increase residual nitrate in the soil. And what is the result of this accumulation of residual nitrate or post-harvest nitrate? The nitrification process can modify the pH of the soil. We saw there is a decrease in soil pH. The electrical conductivity, we saw there is an increase in electrical conductivity, and these two changes will affect the berry yield. Thank you.
Awesome. Any questions for Amy? Quentin?
So on your blueberry results slide, you had rates that were 50 to 100%, 100 to 150%, and 150 to 20. What were those rates based on, and is your rate changing year to year, or were those set amounts? Did these plants start out as little plants, and were they growing?
Yes, in fact, we used the guideline. Yes, and depending on the year, on the age of the plant, we selected on the recommendation guide what is the amount recommended, and the order rate were adjusted based on that.
Just on this the year.
Yes.
Mm-hmm. There was no soil sampling or tissue analysis that you went by.
No.
Is your study going to continue out to 2017?
Yes. Our goal is to keep that experiment as long as possible and see what will be the result in, let's say, 5 other years or 6 other years.
Okay, any other questions for Amy? Okay, please help me in thanking him.